In this presentation, I'll be giving you an overview of the value and challenges of using complex, high-dimensional data in biological research. First, I'll explain what we mean by big data for biologists. Second, I'll discuss where traditional biomedical researchers fit into this new big data world. And finally, I'll talk about some of the general challenges of doing big data science. As biomedical researchers, we're taught to be reductionist, simplify the problem to gain insight into a specific biological situation. However, we understand that biology is complex. Our one cause and effect scenario is subject to multiple direct and indirect signals. Ultimately, we don't want to ignore this complexity, because if we do, we're like the old story of the blind men describing an elephant. Each of us has a limited perspective. We'll just see pieces of the puzzle and not the whole. Fortunately, we live in an amazing time. Before the year 2000, we had only tools to do single gene and single protein research. And because of this, we were often just trying to discover the pieces of the system we wanted to study. But in 2000 came the first published human genome sequences, followed by genome sequences of the mouse and many other model organisms. Although this was just the start of the omics era, it helped us see that data generation itself didn't have to be a barrier to scientific progress. Today we have a huge amount of information at our fingertips. We have databases and repositories for the transcriptome, so Geo and GTEx, for gene regulatory elements, the ENCODE project, proteomics and metabolomics, the PRIDE and HMDB databases, genetic variation in the 1000 genomes, and JAX mouse resources, and specific phenotypes that link to genotype, so TCGA for cancer and JAX and the gene network for mice. Our challenge is, how do we use all this information? Omic data is what is commonly referred to as high-density data, where we have a lot of information generated for each individual sample. Big data is that and more. It integrates multidimensional, high-density, variously formatted data types with the goal of gaining insight into the function or regulation of a biological system. Classically, big data is defined by the 3 V's: variety, multiple types of information; volume, larger and larger scales of data; and velocity, more frequent sampling up to real time. More recently, 2 additional V's have been added: veracity, which is an assessment of the uncertainty, messiness, or trustworthiness of the data; and value, an assessment of the worth of adding the complication of using the other V's. When people talk about big data in medicine, they can mean one of 2 things. If they're interested in biological mechanisms, development of disease biomarkers, or discovering treatments, this is the more omics-driven type of science. This is what we'll be discussing in our course. Alternatively, people use medical records to work towards optimizing healthcare delivery or reducing the cost of healthcare. This is also interesting, but beyond the scope of our course. Some have argued that the explosion in data generation has made the traditional approach to science, where researchers test carefully crafted hypotheses, obsolete. Instead, they argue that the data will speak for itself. While this is clearly an overstatement, the new era of big data does challenge the traditional biomedical researcher in several ways. The biggest challenge in working with omic and big data is that it's complicated beyond what a human mind can handle by itself. Here we're looking at data from a large-scale imaging study where 12 different imaging technologies were applied to the brains of subjects. Each of the 12 imaging methods provides information that's common to all methods, for example, the size and shape of the brain image. But there's also images that may or may not be unique to each method. The problem is that even in a relatively small 7 by 12 matrix, just like I'm showing you here, it's impossible to see any patterns. Because of this, we need to develop a new multidimensional mindset that's aided by new analytical tools. This underscores the fact that biological big data science will require a committed collaboration between multiple disciplines. The generation of data requires the expertise of people who develop and implement high-end methodology for high-density phenotyping. The processing, analysis, and annotation of high-density data requires statisticians, computer scientists, and bioinformaticians to reveal the interesting features of the data. Finally, the role of the biologist is to provide context and focus to the project. This occurs both at the level of the study design and in providing expertise to place the findings in a biomedical context. When these three groups of experts converge to work on the same project, this is when big data has the greatest chance to make an impact. Unfortunately, for most of us trained in biomedical sciences, what you're seeing on this slide is what often happens. We plan our studies, collect our samples, then send them off to service centers where we don't know exactly what happens there. This places a biomedical researcher in an uncomfortable spot of having data and analyses that can't be easily explained, much less verified or trusted. However, many things happen in the black box of big data, and traditional biomedical researchers will have to become comfortable with these processes. Of course, we'll still have to design high-quality studies that address biomedically relevant conditions. These frequently use our collaborations with statisticians. But we'll also have to get used to the fact that there are analytical, computational, and statistical challenges that happen at every step of the process. This includes processing and cleaning data, determining levels of differential gene expression, visualizing output, annotating data, and determining patterns in data. All of this will allow us to better interpret how conditions like lifestyle or disease affect biology. And this will only get more complex as we start combining multiple types of data into our research designs. So what's the secret to making the transition to doing biomedical big data science? Any number of things. Curiosity, persistence, and a willingness to work in teams, just to name a few. The goal of our course is to demystify the big data science approach to research. In the next video, I'll give you an overview of what we're going to learn in the course.